Melanoma is a curious disease, more so than with some other cancers. We see patients who can have very late relapse and patients who have very early relapse. And of course, some patients don't relapse at all. So we've always known that some patients do well and other patients don't. And as you'll know, with melanoma, we're very excited by the fact that the immune system can, under certain conditions, appear to control the disease, and some of the new therapies that we have are proving to be able to stimulate the immune system in a way that controls the disease. The question that we were asking was, without any therapies, whether there was any evidence that the immune system was important in differentiating between which patients relapsed and which patients didn't. So what we did was we took a collection of patients from my institution at Mount Vernon and also my colleague's institution, Christian Ottensmeyer in Southampton. And these were patients who had all had high-risk melanoma resected. So they had either had a primary lesion that was a nasty lesion or had had stage 3 or stage 4 disease resected. So they had no measurable melanoma left. But we knew that because of the features of the resected disease, that a significant number of them will relapse, but there'll be a proportion who won't. And we collected serum, so blood tests for a year, at monthly intervals from that cohort of 43 patients. And what we've done is to look at a panel of tumour antigens. So these are serological responses, so antibody responses to tumour antigens that are expressed on melanoma cells. A company called Serometrics has a technology, and we collaborated with them. So they have an assay where they can look at a panel of tumour antigens. And what we showed was that the patients who had immune responses to those tumour antigens detected by the presence of antibodies against the antigens that were circulating in the blood, those patients over quite a long period of time did not relapse from melanoma. But the ones who didn't have evidence that they had mounted an immune response to those antigens were the ones who did relapse from their melanoma. And the implication is that it may be that the ability to induce an immune response significantly influences whether you do or don't relapse having had a high-risk melanoma. And next steps would be to validate it in a larger data set. We've seen data from this meeting that there are a group of patients who are receiving targeted therapies and immunotherapy who have long-term durable responses. I'd be very interested to know in those groups of patients whether the presence of an antibody response to that panel of tumour antigens influences whether or not somebody's going to have a long-term response to benefit. So those are the obvious studies to do, and we're in discussions about doing those.